Well, good afternoon, everyone. My name is Andrew Pepper, and I have the absolute pleasure of monitoring today's ATI seminar series. First off, we would like to acknowledge that the University of Alberta is located on Treaty 6 territory and respects the histories, the languages, the cultures of First Nations, Métis, Inuit, and all First Peoples of Canada, whose presence continues to enrich our vibrant community. We'd also like to thank Astellas for their continued support of the ATI seminar series. And we're exceedingly fortunate today to have a true tour de force in the fields of islet biology, clinical islet transplantation, and beta cell replacement therapies for the treatment of type 1 diabetes, Dr. Per-Ola Carlsson. Dr. Carlsson is a professor of medical cell biology at Uppsala University, Sweden. He also serves as a senior consultant in endocrinology and diabetology at the Uppsala University Hospital and as the director of Uppsala's Diabetes Center. His research focus over the years has been on islet physiology, islet vascular biology, stem cell research, beta cell replacement, and early phase clinical cell therapy trials in type 1 diabetes. He has authored more than 200 peer-reviewed publications and is the PI on the present first-in-human study of hypoimmune islet cell transplantation in type 1 diabetes. Again, it's an absolute pleasure to introduce Dr. Carlsson, whose seminar is titled Surviving of Transplanted Allogeneic Beta Cells with No Immunosuppression. The floor is yours, and I welcome individuals after to open up their screens and open up their mics and ask Dr. Carlsson any questions they may have. Thank you, Carolyn.
Thank you, Andrew, for your kind introduction. I'm happy to be here on Zoom. Let's see if we can share a presentation then. Can you all see this presentation?
Yeah, we can see it. You may just need to put it into presenting mode.
Yes, great.
Perfect.
So my speaker disclosure to start with, I have financial or relationship with industry since I'm a co-founder and a shareholder of a medtech company called 12 Analytics, working on CGM analysis, but that company was not involved in the present study. I should also disclose that I have served in scientific advisory boards for Sana Biotechnology, which could be relevant for this presentation, but also for the stem cell company Pancreas. And this is, of course, well known, but I give a short introduction since I'm not fully familiar with all your backgrounds. Anyhow, this is the first insulin-treated patient back in 1922 in Canada, Toronto. And obviously, this has been the treatment for more than a century. Of course, we have refined the insulin therapy in many different ways. And of course, the medtech support have improved tremendously over the years, but it is still the same treatment. And while insulin treatment is life-saving in the short term, it does not normalize glucose control. Perhaps it's rather normalized nighttime in many ways if you use modern AID systems, but it's always difficult to control the glucose during meals, during exercise, during stress, and whatever you do during daytime of your daily life. And therefore, these individuals have a shortened life length and also a lot more cardiovascular disorders than other people. So if we would like to cure type 1 diabetes, my belief is that we need two different things. We need access to a large number of cells, but that is not enough since we have been performing, both you in Edmonton and we in Uppsala have been performing quite a lot of islet transplantation in the past, and at least in the Nordic network for clinical islet transplantation, we have a surplus of donor pancreases. We don't even transplant all that we get. Some go for experimental purposes and research, since there are a lot of side effects of the immunosuppressive treatment, like infections, like cancer, like renal failure, and therefore we only transplant a minimal number with severe hyperglycemic events or already receiving a renal transplant. So besides access to large number of cells, which would perhaps be stem cells, we would need to avoid the immunosuppressive treatment to make this a therapy for the majority. So you are all familiar and have probably been involved in the Vertex phase 1-2 trial. I have not been involved in that, but anyhow, it was of course a success with 10 out of 12 of a full dose transplanted that became insulin independent. But it is also a concern that one of the 14 treated subjects died due to the immunosuppression in a Cryptococcus meningitis, and that in some way show this therapy and both the capacity it has to treat type 1 diabetes, but also the drawbacks of the use of immunosuppression. So as mentioned, we have been for many years been transplanting donor pancreases, and we have had success in reversing hyperglycemia all since the Edmonton Protocol published back in 2000. But as of now, I would say that we are actually decreasing the number of transplants also in Scandinavia, since we have this AID system, which makes the indication of severe hypoglycemic events more rare than before, actually. So in order to fully be able to use this progress in the stem cell field, where they have generated then insulin-producing cells that are functional and more or less as good as cadaveric donor beta cells, we need to, in some way, be able to transplant without immunosuppression. It could be encapsulation, it could be immune tolerance, but we have been working with a third concept, which is immune evasion. And that concept is mainly based on the work by Sonja Schreber, a professor originally from Germany, but has been working in San Francisco for many years and is a co-founder then of Sana Biotechnology. And she has, in numerous studies, showed that if she remove beta-2 microglobulin and C2TA, and therefore knock out HLA class 1 and class 2, and at the same time also overexpress CD47, she can, and she has found that she can transplant numerous tissues between individual mice strains without reaction. It has been shown for cardiomyoblasts, but also, for example, for pancreatic islets. And she has also showed in a mouse model that where she had implanted a human immune system from a subject with type 1 diabetes, and then she has, from the same individual with type 1 diabetes, generated iPSC cells that were converted into insulin-producing cells, either with these 3 genetic modifications or wild-type cells. And then she noted that she got an immune attack of the wild-type cells, presumably a recurrence of autoimmunity then, while the triple gene-modified cells were spared. So it seemed in these mice models that also the autoimmunity could be evaded by this strategy. And finally, she used a monkey that was chemically induced with diabetes, streptozotocin, and then she transplanted beta cells from another monkey and were able to reverse hyperglycemia in that monkey without immunosuppression. So based on this concept, I had quite numerous discussions with Sonja, and we decided on performing a first-in-human trial to investigate the possibility to transplant insulin-producing cells between individuals without immunosuppression. And this is a proof of concept study that was performed as an IST at Uppsala University Hospital. And what we did was that we got approval from the Medical Products Agency and Ethical Review Authority to use a donor pancreas from a brain-dead cadaveric organ donor. We isolated pancreatic islets at the Rudbeck laboratory in Ole Kosken's lab. And got rather many islets, and then the islets were sent to Oslo University Hospital, where the islets were dispersed into single cells. They were then genetically modified in these 3 different ways by knocking out beta-2 microglobulin and C2TA, HLA class 1 and class 2, and then overexpression CD47 with lentivirus. Then the cells were reaggregated into islet structures, released as an ATMP back to Uppsala University Hospital. And then we had the opportunity to transplant an individual with type 1 diabetes, which had type 1 diabetes since 1987. We had possibilities to transplant a dose between 25 to 80 million genetically modified cells, and we transplanted almost 80 million cells. We transplanted them intramuscularly into forearm muscle, and that was due to safety purposes. Since we have genetically modified them, we needed to be able to monitor them by imaging. We needed to have a strategy to remove them if necessary. And both imaging and also removal is impossible, of course, at the intraportal site. The donor had a blood type O. He was 61 years of age and he had no diabetes. The recipient, as mentioned, had his type 1 diabetes diagnosis back in 1987. So 5 years of age. He also had blood type O+, and he was 42 years of age at transplantation on December 2nd last year. He had a poor A1C, as you can see, which was also a requirement actually from the Medical Products Agency, that we should target individuals with the highest need of this therapy if successful in the end. We took real care to spread the islets as much as possible, and therefore we created with an IV catheter, the plastic part of an IV catheter, 17 channels, and injected the islet cells slowly while retrieving the cannula. And you can see this in this movie. It's not very advanced surgery, of course, but of course that is also beneficial if you would like to spread this concept. Slowly injecting the cells so that they will not be damaged or build up too high fluid pressure, and on the end put on some cotton swab to prevent leakage. And following surgery, we followed the subject and to start with weekly for safety. Of course, we also looked for a function of the graft. This individual had no C-peptide, no measurable endogenous insulin production at all prior to transplantation. Then we sample it, of course, frequently at all visits after transplantation. We also performed a mixed meal tolerance test at repeated occasions. For safety, we did MRIs, and we also included a PET MRI at week 12, enabled to use an exendin 4 tracer, GLP-1 agonist tracer, to see if we could visualize the cells if they survived that far. We also performed a couple of immunological assays to see if we could evade the adaptive immune system or the innate immune system. And this is data released so far up till 6 months. The subject is otherwise now 9 months after transplantation. And I'm happy to say there has been no serious adverse event in the study. There has neither been any adverse event, possibly or probably related to the study drug in the study. So this far it has looked very nice. With regard to functional data, prior to transplantation, this individual had no measurable C-peptide at repeated occasions. We also performed 2 mixed meal tolerance tests actually prior to transplantation, and at all time points and at all occasions measured, he was with C-peptide below the limit of detection. The sensitivity of this assay was 0.48 picomole per liter. After transplantation, he has had stable values, although low, I must admit, on 10 picomole per liter, but it has been stable at all time points, and of course reflect a very marginal cell mass transplanted, which reflects about 5% of a curing dose. In response to a mixed meal tolerance test, he has increased at all tested time points with about 70% in C-peptide. So this has also been very consistent and with no signs of loss of graft function. With regard to the MRIs, those were mainly performed for safety. Still, early on, we could see some fluid in the T2 stir weighted sequence, which is sensitive to water and fluid. That disappeared rather quickly since it was absorbed after early inflammation, cell death, I guess also, and also some water or buffer we injected with the cells. After a couple of weeks, as this sequence from day 28, we can see white spots that we interpret as highly vascularized islet structures. We could, of course, not be certain of that, but it looks like the highly vascularized structure we have seen in larger grafts, autotransplants previously. This is perhaps more convincing with regard to anatomic signs of survival of cells, namely the PET MRI from week 12. Where we had a 68 gallium exendin 4 tracer that was injected, and as you can see on the right part of the picture, you can see that we have a rise in the standard uptake value immediately after injection of a radioactive tracer, both in the surrounding muscle and in the part we believe we have the graft, and that is of course the arterial perfusion with a tracer, but then it declines over time in the muscle area, while the tracer seemingly accumulate in the region where we have the grafts. We have then performed immune analysis where we have spared some of the donor islet cells and some, and then exposed them at different time points to the patient's immune cells. From the donor, we have saved both so-called unmodified wild-type islet cells. We have islet cells where we only have a knockout of HLA class 1 and class 2, but normal amounts of CD47. And then the triple gene modified cells with knockouts of HLA class 1 and class 2 and overexpression then of CD47. And then these cells were in different wells exposed to T cells, donor-specific antibodies, NK cells, or whole blood from the recipient. And this is a busy slide, but it shows that we have performed these assays at repeated times throughout the study. And what you can see is that for the wild-type islet cells, we have, prior to transplantation, no activity towards the wild-type islet cells from the patient T cells, because the patient T cells have never seen, so to say, the wild-type cells before. But since the graft that we transplanted were a mixture of gene-modified and non-gene-modified cells, so after transplantation, there was a strong immune recognition of a patient's T cells against the wild-type islet cells. And from week 1, you can see that the killing in these in vitro assays are very rapid and full. And that occurs although the spot frequency levels off over time. It's still a very strong activity of a patient's T cells against the wild-type islet cells up to week 26. With regard to the double knockout islet cells, there is no killing out of a patient's T cells. And the same goes for triple gene modified cells. We can also see that for the patient's B cells, the same as we can see for the T cells, namely that prior to transplantation there is no activity. After exposure to the graft, there is production of donor-specific antibodies to start with, mainly IgM, that shifts into IgG over time. As for the T cells, there is no binding of donor-specific antibodies to HLA deficient cells or to the triple gene modified cells. But the double knockout islet cells, although they are spared by the T and B cells, they are rapidly killed even before exposure to the donor cells by the patient's NK cells. And so we can see a rapid killing of donor cells, both at baseline and throughout this time periods up until week 26. And that is, of course, due to the so-called missing self phenomenon described by the Klaus Scherrer back in 1990, which means that if cells have decline, a removal of HLA class 1 and class 2, the cells are considered as strange and will be removed by NK cells or macrophages. And that can happen, for example, with virus-infected cells or cancer cells. So this kicks in for these double knockout islet cells. But for the hip islet cells, we can see no killing by the patient NK cells. And we can also see by adding a CD47 antibody that we can remove this capacity of the hip islet cells to be spared by the NK cell attack. So this is also a mechanistic finding. So to summarize, the wild-type islet cells, after transplantation, we can see very rapid killing of them by the patient blood, and that occurs at all time points up until week 26. For the double knockout islet cells, we also have a very rapid killing that occurs even at baseline and at all time points after transplantation. While for the triple gene modified cells, the HIP cells, we can see no immune attack at any time point. and that can also be visualized in this video, which is a video of wild-type islet cells, double knockout islet cells, or the triple gene modified islet cells, while we then expose those cells to the patient's blood, and then have a fast-forward video over 4 hours. And then we can see that the wild-type islet cells are rapidly killed, but double knockout islet cells are also rapidly killed, whereas the hip islet cells are spared. And this project then was a collaboration between Uppsala University Hospital, which is called Akademiska sjukhuset in Swedish, Oslo Universitetssjukhus, which is Oslo University Hospital in Norway, and of course Sana Biotechnology, which helped us with gene modification protocols, and then the Leona M. and Harry B. Helmsley Charitable Trust, which financed the project. And this has been published this summer in the New England Journal of Medicine. The paper version is, I believe, from September 4th, actually in the same number as the Vertex study is published. Thank you for your attention.
Thank you, Dr. Carlsson, for an incredible lecture and a real pioneering efforts in the field of beta cell replacement therapies. I see Dr. Shapiro has his hand up. Please, Dr. Shapiro, feel free to unmute and ask your question to Dr. Carlsson.
Per-Ola, thank you very much for a wonderful talk and a really terrific scientific experiment with very interesting clinical and in vitro findings, very clear in many respects. The obviously the dosing of the islet cells that were transplanted was very small. Just by necessity, you had to dissociate the entire human islet prep into single cells to transduce it and then reaggregate it, and then you place the islets obviously in an ectopic site. That meant that only a small fraction were to survive. So we're probably only seeing a fraction of the potential of this approach. Difficult to predict how good it's going to be over the course of time. One of the concerns I have is that you didn't mention this, but I'm aware of the data that the only about 20% of the cells are actually transduced. So 20 to 30% is transduced. That means at least 70 to 80% of the cell load that you transplant doesn't have any immune modifications in it. And my worry is that you would sensitize the recipient and prevent them from receiving further islets or kidney or anything else down the road with that kind of approach in an unmodified manner. So do you have any data on the panel reactive antibodies in your single case and what that risk might be to future patients?
I think it was 40% about that was fully gene edited, but definitely we transplanted a lot of non-gene edited cells. We could not see actually, we did look at the HLA immunization. We haven't seen actually so much of that. And this far we have only measured with regard to the autoantibodies, GAD-IA2 and SINC-T8, and both of them were the same, both prior to and after transplantation. Of course, this is definitely a concern that it could be a problem if there would be a need for a clinical transplant in the end. But with regard to this concept, if we would transplant more of these cells, I think it would be no concern because it's HLA deficient cells.
If you continued to use human islets, though, presumably with the cell losses, with the disaggregation and reaggregation, and then the ectopic site, and you mentioned you'd need... then the risk of sensitization goes up quite a lot, does it not? Yeah.
Yeah. So I would say this is a subject that has a poor A1C, but he has no classical indication actually for an islet transplantation. But I think this is a proof of concept more or less for that the gene modifications seem to work. In the end, and what we aim for now, of course, is to use stem cell-derived insulin-producing cells instead. And then you can gene modify out of a stem cell level. And then, of course, we would have all daughter cells genetically modified in the same way. So then we will not have a mixture with non-gene edited cells, which is, of course, very beneficial for a future therapy. And also, of course, that we would be able to generate much more cells. Because, as you say, there was a dual problem. We did not have approval by the MPA to transplant more cells, but even if we would have, we couldn't have transplanted much more cells.
Thank you very much.
Wonderful. I see Dr. Holland's hand is up and then followed by Dr. Helpin.
Yeah, I just, can you hear me? Is the sound okay? I just was wondering about, I was looking at CD47. I don't know anything about this. I see that it's a don't eat me signal, and that it interferes with macrophage recognition as well. Do you know whether this is really an NK effect or whether this is mediated by other innate immune mechanisms, which could also recognize the cells with missing HLA?
Yeah, in the immunoassay, we have tested and showed on the NK cells because we have just purified NK cells and looked at to see if they mediated this immune attack. But you can actually see the same also for macrophages. So it's a mixture attack by both NK cells and macrophages that contribute to this innate immune attack. So the CD47 protects from both of these cells. And as you say, it's an eat and don't eat me signal. There has been some reports, mainly from Australia, that has suggested that knockout of CD47 would improve insulin exocytosis. We have performed the opposite then, obviously, to overexpress CD47, and we haven't seen in the data any decline in function, but that is, of course, something that is of interest to look into further in the future too.
And anti-CD47 has been, there's been worried about its effect on red cells and potential anemia. So have you seen any, you said you don't have any serious adverse side effects, but it's not necessarily a completely safe drug, is it?
No, but overexpression of CD47, it's also possible to have a reversal of that if needed, because we had the implant in muscle, and then of course could do surgical resection, but there is also possibilities to administer CD47 antibodies, and in the experimental studies that has fully cleared the graft. So that is a rescue pathway if needed, if you would need to get rid of a graft for any reason. That is very effective in killing the graft. And those drugs, they are not approved drugs, but they have been in phase 3 trials, so they are rather well studied.
Thank you.
Dr. Helpin, you're more than welcome to ask your question.
Thank you so much. Thank you for this beautiful talk. It's such elegant work. I'm in the HLA lab, and I also just want to say that, like, how proud I am that the first question is an HLA question from one of our clinicians. So it made my heart very happy. So I was going to ask about the PRA, but James beat me to it. But my other question, and I looked at your paper as well, and so I apologize if I've missed it, but my understanding is that there's still some presence of class 1, HLA class 1 expression. Is that correct? Just like not very much?
No, it should be more or less none.
Like I think it says like 86% or something in your paper absent.
Yeah, okay. Now I know
Yeah.
what you mean, but it is not on a single level. It is not on the single cell level. So there are some cells in the mixed preparation that were not genetically modified, and those were killed within the first week, and that contributed to the sensitization that made the B and T cell attack and the memory that you could see in the slides. So, of course, when we genetically modified them, it's of course more or less impossible to be able to genetically modified all cells in the preparation. So I think it was still a very good success to have 86%.
Oh yeah, that's for sure.
Genetically modified. So, but that meant, of course, that and we did not purify so that we only transplanted genetically modified cells, because then we would have even more cell loss. So we transplanted a mixture of non-genetically modified cells and fully gen-modified cells, and that of course also contributed in the immune assays, but it could of course also have contributed to that we had more initial cell death because of more strong inflammation in the graft or early on.
So do you know, because maybe it was in a supplemental data that I didn't see, but how well matched the donor and recipient pair were in the case study that you presented? Because I'm sure you knew the HLA typing where, because as you said, it's a lot to ask for it to be 0. And so I think it was an excellent success of what you did. But I'm just curious if they were, how many mismatches there were, if you looked at epitopes, maybe there was, and maybe that could be a future component of your work, is to say, to select, or maybe you did that to select, based on mismatch.
I know it was not selected based on mismatch. It was a random donor, and I wouldn't say it was random recipient, because we had a waiting list of different recipients in different blood groups, and since it was a donor with a blood group, it could of course have been any recipient, but it was perhaps the one that had been on the waiting list the longest, that was more logic to take. So and there was of course HLA mismatch, but it was of course it could have been worse.
Yeah. And I guess because there are certain class 1 and class 2 antigens we know are associated with development of type 1 diabetes. So that might be also an interesting question, is were any of those antigens present in the donor? Does that make a difference in the short or long term? Anyway, it's a beautiful model. So I really enjoyed your talk, and I think there's a lot of, I'm sure there's a lot of great work to come.
Yeah, and of course, this is one individual. It was a very classical individual with subject with type 1 diabetes with classical autoantibodies and so on. But of course, we cannot be sure that there are subgroups or with some special form of autoantibodies, for example, that could not function in this system, of course, and that will of course be tested in the future. But this is I'm very optimistic based on the subject, but it's still only one subject.
Brilliant. I'm really glad that you mentioned about the CD47 and the potential role in insulin signaling, and that you're looking at that. You don't see a hindrance in insulin secretion, the overexpression. I wonder about beta cell exhaustion over time, and maybe that could be attributed to graft attrition, not just loss from a recurrent immune activation. But it's nice, at least you don't see in vitro any hindrance of overexpression of CD47. I'm curious how you foresee this approach potentially to expand patient inclusion. You mentioned that this wasn't a typical individual that would be deemed eligible for a classical islet transplant, but we know the benefits of just having some islet function and some C-peptide function. Is this possible to, without the risk of prior sensitization or future sensitization, of providing some metabolic benefit of even having a subtherapeutic dose to these individuals?
Yeah, so this individual perhaps mainly he has no problem with hypoglycemia. He tends to stay on the high scale of glucose values. We actually improved the metabolic control quite much after his transplantation by improving his insulin regime, but it's still not perfect, of course. It's difficult to say that he would be an islet transplant candidate with immune suppression, but hopefully I would love to be able to include him in some future trials also, because this is of course something that he had no much benefit of himself, since it's only a 5% of a curing dose. So it would be I would be extremely happy if I could have in the future the possibility to give him a more or less curing graft.
Excellent. And you mentioned about it was fascinating about the intramuscle transplant route that you did, and you mentioned about the regulatory considerations of having retrievability and graft monitoring. When you had 17 sites for about 5% of the beta cell mass that was transplanted, have you thought about scalability to get to a curative dose? Because rough math, that would be hundreds of injection sites. We thought about how you would use the intermuscle site as a clinical translatable site for a full therapeutic dose.
Yeah, so I think we were a little bit, we transplanted the cells in 3.2 milliliter, and perhaps we used too much buffer actually. Just calculating on it, I think we could even transplant 1 billion cells in about, I don't know, 7-8 milliliter if you take away most of the buffer. And the difficulty is mainly to have them as a pearl on a string fashion, and the major strategy I think is to use longer needles actually, or longer plastic parts of IV catheters, so that we have longer channels instead, because that will save time instead of creating numerous more channels.
Right, and also seeding density may be an important concept, of course, with your islet vascular biology. And maybe it was fortuitous that you did have a lot of tracks and having a lower seeding density per
and we
individual
should be
track.
aware of, since we transplanted a lot of non-genetically modified cells, well, it was 80 million cells, but it was, of course, much more cells to start with if we also consider those non-genetically modified cells.
Right, brilliant. I don't see any other questions in the chat or any other hands raised. And if that's the case, I'd really like to thank Dr. Carlsson for your really stimulating and pioneering work and presenting with us here today at the ATI seminar. And I would like to remind the rest of the community of next week's seminar. Please join us on Wednesday, November 5th, as we host Dr. Adam Feinberg from Carnegie Mellon, who will be presenting on the 3D bioprinting of human tissues and the path towards translation. And I believe Sean has put a link to that in the chat. So thanks again, Dr. Carlsson. Hope you're healing well. And again, congratulations on your success with the clinical trial.
Thank you. It has been a pleasure.
Thank you so much.